rapid, it's very effective, it's non-specific, and lacks immunological memory. And then after the innate immune response gets saturated through antigen presentation and T-cell activation, you get adaptive immunity, which takes longer and is very specific and builds immunological memory. And so this is the traditional paradigm. But if we kind of zoom into what's going on a cellular level, so on the left is the innate immune response. So there's a pathogen invading an organism. And so initially they encounter phagocytes that then try to clear, for example, in case of this current pandemic, that SARS-CoV-2 virus. And when that system gets saturated through antigen presentation, what you see on the right, this paradigm has been challenged, because if you kind of look at what's really going on at the level of innate immunity, there is some kind of primitive memory function, which is referred to as trained immunity, because once myeloid cells encounter a specific pathogen, they can undergo metabolic and epigenetic rewiring, and then they're basically more aware or more alert. So this is not priming, and because they're still at a ground state, but on the epigenetic level, there's a increased responsiveness upon encountering a unrelated stimulus or pathogen. And so this is a concept that's been discovered by Mihai Netea, who we collaborate with very closely. And yeah, this kind of like, I won't go through all of this, but this kind of captured the essence of trained immunity. And so if you look at different processes ongoing in particularly myeloid cells, and so on the top, you see these pathogen recognizing receptors. We see the TCA cycle. On the bottom, you see epigenetic activity, so histone modifications and these type of things. So if you kind of zoom out and put that all in context, then through metabolic rewiring, upon encountering a pathogen, as through these pathogen recognizing receptors, there's epigenetic changes. And so this could be also considered a very primitive form of innate immune memory, which is referred to as trained immunity. Well, and so trained immunity, I'm a biomedical engineer, a chemist, undergraduate in chemistry, and then a PhD in biomedical engineering. So we create technologies. And so when you use this framework of trained immunity, then suddenly there's a lot of stuff that you can accomplish. So when we started working with Mihai, that kind of opened up our eyes, or looking at the possibilities, to kind of take control of the innate immune response and of the entire, in that way, potentially of the entire immune response. So this is something that, we're very excited about. And then if you look at all the molecular processes at play in trained immunity, you see, I mean, I'm not going through all of these, but there's lots of possibilities for interventions. Okay, so this all applies, what I've just discussed, all applies to individual myeloid cells. Given that monocytes have a lifespan of a couple of days, that doesn't sound like trained immunity is a durable functional state of the immune system. That's something that at least appears to be on first glance to be something that is specific to myeloid cells. And so they have a short lifespan, so this is something that wears off very rapidly. Well, that's not the case, obviously, and it's because trained immunity is kind of adopted by particularly progenitor cells in the bone marrow. And so they adopt this trained immunity, that's called trained immunity phenotype, or this trained immunity functional state. And they start producing myeloid cells that by themselves are trained myeloid cells. And so the induction of trained immunity, at least from our experience, induces myelopoiesis. And these myeloid cells, they have the trained functional state. And so these myeloid cells are hyper-responsive. And in mice, this lasts about 6 to 8 weeks. In patients, this is observed up to 2 to 3 years. Anyway, so there's all kinds of signaling involved, but we can hit the bone marrow with with nanoparticles pretty easily. And so that's our strategy. So if you didn't really pay attention to everything I've told in the past 10 minutes, then this is what we actually want to accomplish. And so we want to target innate immune cells, antigen presenting cells, to take control of the entire immune response, which also has implications, obviously, through antigen presentation on the adaptive immune response. And so what we've built is nanotechnology in order to reach these cells and to modulate their behavior or reprogram their behavior, so you will. We integrate imaging to visualize, what's actually going on. So we've built all kinds of immunoimaging methodologies, particularly based on PET-MR. And then this talk, I'll focus on cardiovascular disease and the workflow that we've put in place to go from mouse models, but also large animals. And there, particularly in these large animals, imaging becomes very important. Okay, so I don't think this is an audience that needs to be educated on the pathophysiology of cardiovascular disease, but obviously for a long time this has been mostly looked upon as a lipid-driven disease and the accumulation of lipids in the vessel wall, ultimately causing vessel wall rupture that can cause ischemia, like a myocardial infarction or a stroke or renal ischemia, peripheral artery disease. But very nice work from, for example, Phil Swirsky and Matthias, who's also on this call, and others, Esther as well, that have investigated the role of the immune system and the inflammatory response in this disease process. And so something, briefly about atherosclerosis. So what's the technology that we then develop, that we focus on to engage the innate immune cells? We use nanotechnology. For those who are not familiar with nanotechnology, that's typically things that are in the range of, let's say, 10 nanometers up to a couple of 100 nanometers. That in the field of nanomedicine, that's typically considered a nanoparticle. There's a bunch of definitions. I won't go through some. Some definitions say that it has to be smaller than 100 nanometers in every dimension. But in the field of nanomedicine, we've also created materials that are up to, let's say, 500 nanometers. So that's a half a micrometer. And so that we're working in this range. And traditionally, nanomedicine, the prime purpose is to increase the therapeutic index. And so a lot of, for example, cytostatic agents are highly toxic. And so the idea is if then you encapsulate them into a nano formulation, you can improve delivery to, for example, a tumor, and that allows you to decrease the dose, and that increases the therapeutic index. So that's traditionally the field of nanomedicine. And that requires one thing: you have to evade the immune system. The immune system, reacts, pretty vigorously to whatever is administered, particularly in the nano size range. So there's been developed all kinds of strategies since the late 80s to basically equip nanomaterials with kind of shielding. It's also called stealth technology. And in this case, what you see here on the screen are these polymers that are coating the, in this case, the liposomal nanoparticle. And it's all designed to evade the immune system and to get good accumulation in for example, a tumor. And this works. And this is our patient data, where you actually see there's a lot of tumorous lesions in this specific patient, and you see the accumulation of long circulating liposomes into these lesions. However, if you look at what is being produced in the field of nanomedicine and what actually leads to a tangible product, it's very disappointing. And so, and you see all kinds of strategies to increase the complexity of these nanoparticles, equip them with all kinds of different modalities and other functionalities. But yeah, the amount of medicines that are actually developed clinically and that are FDA approved is very limited. So looking at this, it's because probably the targeting concept and the drug delivery concept is maybe preclinically it works, but it's not as potent as the field would have wished. But we don't care, because we actually embrace the fact that these nanomaterials have inherent interactions with innate immune cells. So instead of seeing this as a disadvantage that we try to evade, we actually embrace the fact that these nanomaterials can be exploited for immunotherapeutic purposes. And so now it's not about drug delivery, it's about engagement of the innate immune system. So what do we use? We kind of plagiarize nature, where we look at specifically one protein, apolipoprotein A1, which is the main protein constituent of high-density lipoprotein, as everyone on this call should know as good cholesterol. We don't care about cholesterol too much, but we like the interactions ApoA1 has with immune cells, with macrophages through ABCA1, ABCG1, and scavenger receptors. And so this concept is a natural targeting concept that naturally occurs in our bodies. And this is a nice concept that as a biomedical engineer, you would like to plagiarize. So looking a little bit closer to the morphology and the buildup of a lipoprotein, you see that there is a hydrophobic core, there's a monolayer of phospholipids, and there's these apolipoproteins integrated. And what we essentially do in this conceptual, what I'm going to show next, and so this is not what we actually do, but then you kind of grasp the concept, is that we extract blood. In the lab, we actually get blood from the blood bank, or we get HDL fractions, and then through very extensive centrifugation procedures, then we can collect the HDL fraction, which is rich in ApoA1. In the lab, we disassemble these nano formulations, then we put in, the payload, and then essentially you end up with a nano Trojan horse. That's the concept. This is not how it works in reality. Oh, sorry. In reality, we use this type of technology. So most focus on microfluidics technology, where we then have as the main constituents of these formulations are obviously apolipoprotein A1, phospholipids, maybe cholesterol, and using different solvents and different mixing rates, etc. They can actually generate spherical and discoidal nanobiologics. So now we don't call them HDL anymore, because we've been playing with them too much to really say that this is still HDL. But we can make these things spherical, and we can make them discoidal, and we can do this in the lab, and we have this nice specific interaction with myeloid cells, and we also see with progenitors in the bone marrow. And I'll show you some data soon. So this is a cryo electron microscopy, where you can nicely see that these spherical particles are about 40 to 50 nanometers in diameter. These discoidal ones, they're much smaller, they're about 15 nanometers. But it's not all that we've done. We've actually created a large library of what we call nanobiologics. So these are ApoA1 based nanomaterials that have two essential building blocks, phospholipids and apolipoprotein A1. And by the inclusion of other building blocks, molecular building blocks, we can play with the morphology. So we can make these things really small, and they look like discs, and we can grow them up to 400 nanometers, and then they're nicely spherical. And then we, for example, have to include polymers. So there's all kinds of tricks to generate these type of libraries. So to zoom in, so what you see here, these are discs. And so this looks like a stack of coins. I hope everyone can appreciate that. And then we can include a hydrophobic payload, and they become spherical. We can include these polymers, and we can grow these things up to 400 nanometers. All right, so then we have that library. We can radio label the individual nanobiologics from this library with, for example, zirconium-89, which is a radioisotope with, as a physical decay half-life of approximately 3 days. But we can also fluorescently label them, so that we can subject that nanobiologic library to, in this case, apolipoprotein, ApoE knockout mouse, so a mouse that has atherosclerotic plaques. And so then we can look at biodistribution, we can look at the vasculature, we can look at cellular specificity, and then, based on our observations within that library, we can select a specific material. And these data look something like this. So this is a very busy slide. I'm not going over all the details, but what you see on top are different aortas from ApoE knockout mice that have advanced lesions. And then you see we use, in this case, near-infrared imaging to look at the distribution of these nanobiologics in these aortas. And we can look at, other organs. This is on top, what you see is based on optical methods. On the bottom is based on quantitative radio labeling and then performing gamma counting and autoradiography. And so the same we can do on a cellular level with flow cytometry. Again, it's not about the details, but I hope you're able to appreciate that within the library, this is differential behavior. So some materials are more prone to be taken up by macrophages, others tend to have a higher propensity towards monocytes or dendritic cells. So as you see, there is this diverse behavior within that library, and depending on what process that we would like to target or cell type, then you can select from this database or from this library. Okay, so imaging is an integral part of what we are doing. That's related to, because my lab has always been part of what used to be an imaging institute. It's now called the Biomedical Engineering and Imaging Institute. So still imaging is very important at the institute. So we have nice access to preclinical imaging equipment, but also clinical imaging equipment that allows us to scan large animals. Yeah, this is an overview of typical imaging modalities that we like to integrate and are used a lot. So for example, CT imaging, that's a very popular method, particularly clinically. MRI, in the context of cardiovascular disease, MRI may be even more powerful because you have this nice soft tissue contrast. We use optical imaging methods, and I became a fan of particular nuclear imaging, PET imaging. And that's very useful. So some of these techniques like MR and CT are anatomical imaging techniques, and they give you information on the inside of the body. Optical imaging and PET imaging are tracer technologies. So you look at a specific tracer that you then can visualize. So these techniques have to be combined with anatomical techniques. Yeah, so Carlos Perez Medina, who started as a postdoc in my lab maybe 6 or 7 years ago, he's a radiochemist, and so he started working on developing all these different radio labeling procedures for these nanobiologics, and that allows us to visualize them. And here's an example of a study that we published a few years back, where we then radio label our nanobiologic injected in different species. So we've done it in mice, in rabbits, and in pigs. Here are typical examples in mouse. In the middle, you see a rabbit. On the right is actually a non-human primate. And so one of the things you're able to appreciate in non-human primate is that the bone marrow is pretty hot. Liver is always more hot, and there's more uptake in the liver than any other organ, but you can nicely appreciate the bone marrow lighting up as well. Yeah, so imaging is useful to look at the biodistribution or distribution of these nanobiologics after we radio label them, but imaging is also very useful as a immunoimaging modality. And so we want to understand the in vivo behavior of these nanobiologics, how they circulate through the body, where they accumulate, how long they stay in the blood, etc. But then we also want to be able to measure, the immunological processes that are maybe changed as a result of our nanotherapeutic interventions. So this is an example of a study where we used multimodal imaging, and we used different types of MRI. We also used nuclear imaging techniques to kind of probe myeloid cell dynamics in the context of atherosclerotic disease and myocardial infarction. This is work done by Max Sanders, who used to be a PhD student in the group. He's now a practicing physician. He still has to do a lot of training, obviously. And he'll soon defend his thesis. And this was his, I would think, maybe his nicest study from his thesis. And just to show how well this works, this is technology, nanotechnology that we build, that we then can administered to these animals. And then there's a specific imaging called fluorine imaging, a fluorine MRI, doesn't really matter what this is. But on the left, you see a control mouse, and then mice in which a myocardial infarction was induced through coronary artery ligation or through ischemia reperfusion. You can nicely see that the spleen signal and the signal on the spine, so the bone marrow and the spleen, is depleted because of the egress of immune cells from these organs. And that's something that we can probe with these type of technology. So that's also why it's very useful to integrate imaging in these type of studies. Okay, so but now let's go to the core of this lecture or presentation, is that we've been applying nanoimmunotherapy to treat cardiovascular disease for a while now. And now I'm going to just go over a couple of studies that we've executed. Some of our multiple studies, but they kind of tell one story. Yeah, again, we're schematically, so the idea is that we have a nanoimmunotherapy that is anti-inflammatory or immunoregulatory, or whatever you want to call it. We inject this. I made a big fuss about the bone marrow. Here we focus on an atherosclerotic plaque, so we kind of zoom in on a coronary artery. You see that it's full of plaque. Then these nanobiologics circulate throughout the body. So they accumulate in the hematopoietic organs, but they also accumulate in these plaques. And then they can resolve the plaque on a whiteboard animation. I'll show you with data that that's actually something that we can accomplish in mice, but also in larger cardiovascular models. Yeah, so in trained immunity, the mTOR pathway was identified by me and Netea's group as a key regulator of trained immunity. And so the natural thing then to do is to use mTOR inhibitors, so rapamycin, and particularly target myeloid cells to see if that can inhibit or blunt the induction of trained immunity. The in vitro assay of trained immunity looks something like collecting PBMCs from a donor, then stimulating these PBMCs with a trainer, which can be BCG or can be beta-glucan. And then you wash these cells after 24 hours, you let them rest for a couple of days, and then you re-stimulate these cells with LPS. And so in cells that are first trained or are subjected to BCG or beta-glucan or any of these other training agents, you'll see increased responsiveness upon re-stimulation. Okay, so what you can do is during the training phase, you can incubate with the nanobiologic, and then you can appreciate that at the level of cytokine excretion, but also at the level of the histone marks of the same cytokines, they can see that the cells that during the training phase are also incubated with our mTOR nanobiologic, they basically behave like baseline cells. And so in the red dotted line are normal cells that then are exposed to LPS. In white, you see the cells that are first trained, and in black, the cells that are treated. That's where you see that the dark bars are very similar to that dotted red line. Now, and so we've applied this technology. I'm not going to discuss this study, but we've applied it to prevent organ rejection in a mouse model, and it works spectacularly well. And so if you, it's very short intervention within the first 5 days after organ transplantation, and then in a subset of animals, we actually can induce tolerance. So these hearts continue beating in this specific model. But we're focusing here on cardiovascular disease, although heart transplantation is a cardiovascular. But here I want to highlight what Mandy van Leent, who's also a PhD student in a lab in New York, has worked on. So she applied this mTOR nanobiologic and another nanobiologic is S6K1. And S6K1 is in the mTOR pathway. I believe it's a little bit downstream of mTOR, but it's in the same pathway. So we developed 2 nanobiologics, one targeting mTOR, one S6K1. You can nicely see that in a training protocol, which is more relevant to cardiovascular disease, where the training stimulus is not beta-glucan or BCG, but it's OX-LDL. and then do re-stimulation, you can nicely see that our nanobiologics prevent the induction of trained immunity. And then when you apply these in mice, and this is actually work done in Matthias's lab, the fluorescence molecular tomography on the bottom, but what you can nicely see is that there is a reduction of inflammation. So these mice have been put on a Western type diet for 12 weeks, and then they receive 4 injections of these nanobiologics in 1 week. But you can nicely see that the plaque inflammation resolves, which you see in vivo by FMT-CT imaging, but also histologically. And by flow cytometry, you can nicely see that there's all kinds of myeloid cell subsets that are significantly decreased in the plaques of these animals. Okay, so this is it for me in that I think now it's enough. You know, I'm a technologist, but one of Mandy's supervisors, Rafael Duivenvoorde, was actually spearheading this study. He suggested to Mandy to open up a Pandora's box, essentially, because in collaboration with Ed Fisher's lab at NYU, we've done laser capture microdissection and then RNA sequencing on the macrophages from these plaques. And, oh, give me 1 second. Sorry. Okay, 1 second. Fuck.
Hopefully he won't shoot the dog.
Okay, you always hope that doesn't happen when you're giving a lecture online, but sorry about that.
Dead? Is he dead?
No, the person who rang the doorbell should, anyway. All right. Hey, so we...
The dog didn't like your spending.
No. So Mandy and Rafa really wanted to, start digging into these data and see what's going on. So I was happy with, okay, we see plaque regression, but Mandy and Rafa then said, well, let's do all kinds of hub analyses. I'm not an expert here, but in both the mTOR nanobiologic treated animals and the S6K1 treated animals that were treated with the S6K1 nanobiologics, we found one very interesting gene, and you may be able to appreciate that is PSAP. So in both of these, in the middle section, I don't know if you can see it, but okay. Anyway, this gene, this is a gene that codes for prosaposin. So then we've advanced this by staining for prosaposin on these sections. There's also very little known about prosaposin's role in cardiovascular disease. So they started investigating this. I'm not going to go into the details of this very extensive study. It's currently being considered at a journal, very nice journal. So we hope we can get that published, and I stimulate everyone to read this. But this was really a expedition, I would say. So we start with nanoimmunotherapy and then opened that Pandora's box, spent a lot of money, but actually got very spectacular data. And this is the main, I would say, in Mandy's thesis, upcoming thesis, this is her main chapter. That's what she worked on a lot. And this is that prosaposin. So, maybe other people should look into this specific protein in this gene. Okay, so that nanobiologic, the mTOR nanobiologic therapy, we then also applied in non-human primates. And so because we can radio label these things, you can nicely look at the biodistribution. So again, like you see the spine lighting up, you can like quantitatively or semi-quantitatively assess biodistribution. And we also found, and we see in the bottom panels, you can also nicely see that it's very well tolerated by primates. So what we really try to do is, be really translational and also, venture into applying this in large animals. And so these are not efficacy studies, and these are PK, pharmacokinetic and safety studies, but it's very valuable to see that it's well tolerated in non-human primates. Okay, then this is work that we've done with Esther actually. So I think most of you are familiar with her work on CD40 ligand signaling, and then, you know, her work revolves still around CD40 ligands signaling, but she started working with knockouts and with antibodies, and ultimately identified that something downstream of CD40, which is TREF6, is a very interesting therapeutic target. And then she identified that small molecule that can be nicely integrated in our nanobiologic platform. And so the idea here is that we target the antigen presenting cell and by that prevent co-stimulation. So this is a study that we published, I believe, 2 years back. Study design. So we've included mice, obviously, for biodistribution work. Also here, we looked at non-human primates. And then in ApoE knockout mice, we looked at therapeutic efficacy, also at the mechanism, and a little bit of safety data. So what we typically observe for these nanobiologics, that they tend to accumulate in atherosclerotic aortas. They also accumulate in hematopoietic organs, and that's what we expected. This is again fluorescence molecular tomography done in Matthias's lab, where you can nicely see that same type of treatment regimen. So we put these animals on a Western diet for a prolonged period of time, you know, somewhere between 8 and 12 weeks. Then they get advanced lesions, which you can appreciate that there's a lot of inflammatory activity, so protease activity. So then we treat these animals for 1 week, so they get 4 infusions of that, in this case, that's TREF6 inhibiting nanobiologic, and you can nicely appreciate the reduction in inflammation, which on the right you see is also confirmed histologically. And so this works really well. Yeah, then we also wanted to apply this in non-human primates. The nice thing about the setup at Sinai is that we have an integrated PET-MR scanner. So MR, I think, is very powerful imaging, anatomical imaging modality. And these data don't show anything except for what you can do with MRI. And so we were interested in the PET signal because we want to look at the biodistribution of these nanobiologics, but, you know, concurrently you can also extract these type of data because we have full 3D data sets, MRI data sets. So it allows us to generate these rendered images. But this is what we're actually interested in. So how do these nanobiologics behave in vivo? Yeah, and then you see that it's kind of like the behavior is preserved across species. That's very similar to what we see in mice, although the kinetics may be slightly different. So typically these nanobiologics, they circulate for a longer time in larger species. Okay, so this is something that, you know, we started maybe in 2008 or 2009 with a PhD student called Jun Tang. And that led to the publication on the which is shown on the left. So these a publication about, we created again, we call them here a nanobiologic that contains simvastatin. And so simvastatin, everyone knows, is a lipid lowering drug, but it also has these anti-inflammatory pleiotropic properties. So we thought, like, if we reroute simvastatin to myeloid cells and to macrophages, we can maybe, you know, really amplify or exploit the anti-inflammatory properties of simvastatin. So that led to the 2 publications. These are very extensive studies in mice. You know, I believe it's about 500 mice per study. And then again, it's very similar story. We see nice inflammation reduction on fluorescence molecular tomography, same regimen. And so 1 week, 4 injections. And so there's very rapid reduction in inflammation. And in this specific study, we saw almost a almost all macrophages were almost completely wiped out from the vessel wall. And that's what we saw histologically. Then a follow-up study, we wanted to know what was going on. And this is, you know, very closely related to Phil Swirsky's work, where he saw that in advanced atherosclerosis, proliferation of these macrophages actually dominates plaque inflammation. And so we started studying what is exactly going on here, and we actually found that our simvastatin loaded nanobiologics, they very efficiently inhibit the proliferation of macrophages in the plaque. And that's how we achieve a very significant reduction in the inflammation of these plaques in mice. So this is what we do in mice, but then we wanted to scale this technology using 3 models. So the ApoE knockout mouse model, that's our regular mouse model, and New Zealand white rabbits, and a pig model. We published these results back in September. Yeah, and this was a disaster in some ways, because we didn't realize what we signed up to when we proposed in a big grant that we were going to do studies with nanoimmunotherapy in non-human primates. That's something we definitely underestimated when we were putting together the application itself. But luckily, we had some very good postdoc at the time, Francois Fay, who's now assistant professor in Paris, who worked on scaling up the technology. And so you have to understand that an average mouse, what is about 25, maybe let's say a very fat ApoE knockout mouse is 30 grams. These pigs are about 60 kilograms. So that's 2,000 mice in one pig. It doesn't scale linearly, but we had to produce tremendous amounts of nanobiologics, and Francois developed that. There was a new procedure, because it's not like a regular benchtop method that we use. We use a microfluidizer system. This is one dose that goes into a pig. And then we've performed a small mouse study, because we want to make sure that the effects of this nanobiologics, which are manufactured through a different procedure, are very similar to what we found previously. And so once we corroborated that, we advanced it to the large animal studies. And Tina Binderup, she used to be postdoc in Copenhagen, who worked in my lab for a year. And she took the lead here in the logistics of all these experiments. It's quite, well, we call that a tour de force. She's very organized. She's a very brilliant project manager. So this is our PET-MR system. And then you can generate these type of data. So this is about biodistribution. This is not PET-MR, that's actually PET-CT, what you see on the left. So on the left, you see rabbits and pigs. So for whole body imaging purposes, PET-CT is actually very valuable. But if you want to zoom in on the vasculature, and in the rabbit, it's in the abdominal region. In the pig, it's in the upper legs, they're the femoral arteries. That it's nice to use PET-MRI, because with MRI, you get that nice soft tissue contrast, and you can look at the vessel wall morphology. And so that are these type of data. So we use PET-CT as whole body imaging, and we look at the targeting of these nanobiologics, the plaque targeting by PET-MRI. You can nicely appreciate in rabbits that there's accumulation in the vessel wall. And imaging protocols, they're developed by Zai's team, and I would like to specifically mention Claudia Calcagno and Phil Robson, who've worked on and worked with us always on all the studies that we do. So all these imaging protocols and beautiful data that I can present here, that's because of the work of these people in Zai's team. And then ultimately, we wanted to probe therapeutic efficacy of this Simvastatin-loaded nanobiologic. This Rafael Duivenvoort is a medical doctor that who co-supervises Mandy. And so he spent a year in my lab as a PhD student, and then he came back as a fellow for half a year. And so he was also very intimately involved in this study and the study design. So we needed surrogate markers for inflammation, for proliferation, maybe for immune cell recruitment. So we kind of used a combination of imaging techniques that kind of, not exactly, but kind of probe these processes. So we use 3D anatomical MRI, which is just to look at vessel wall morphology. We use FDG PET MRI for inflammation. We use 3-dimensional dynamic contrast enhanced MRI that tells you something about plaque permeability, and so that's a measure for monocyte recruitment maybe. And FLT PET imaging can be used for the proliferation of immune cells in the vessel wall. There's 2 radio tracers here, so this is a 2-day scan session, and there's 2 sessions, consecutive sessions, Then we treat these animals. In mice, we do this in 1 week. We want to take it a little bit easy on these large animals. So in 2 weeks, they get 4 injections. And then again, they're subjected to that scan session, and then we sacrifice them. Yeah, I won't go over all the data, but we were struggling a lot with what do we do with all these data. It's a limited amount of animals. These group sizes are relatively small, but we have tons of data. And then ultimately we involved a biostatistician, and she basically said, why don't you just take every parameter that you measure at baseline and then at the end of treatment, and then just accumulate all of that. So these are the individual measures. What you see here is FDG PET imaging, FLT PET imaging, that's a proliferation. You have a dynamic contrast enhanced MRI, and on the right you see regular anatomical MRI of, you can visualize plaque morphology and vessel wall thickness. But if you take all these parameters and then you compare placebo-treated animals versus the Simvastatin-loaded nanobiologic treatment, they can see that in rabbits, 85% of the parameters in the placebo group increased versus 74 versus only 26% increase of parameters in the treated group. And so 74% of the parameters decreased in the treatment group. And in pigs, yeah, it's just a coincidence that they're kind of inverted. But so you see much more parameters going down in the treated animals versus the placebo-treated animals. And so I think this really shows and emphasizes the value of integrating non-invasive imaging and to extract, meaningful data from these type of studies. Okay, so to summarize, so we build nanotechnology that we refer to as nanobiologics that allow us, to regulate specific immune responses. I've shown only data here where we inhibit these immune responses. We can actually also induce these immune responses. We have a paper coming up in about, I hope, a month or something like that, where we show this to be very effective in cancer. So we can induce trained immunity as a modality, as a new immuno-oncology modality, I would say, that works really well. It's very effective, particularly in combination with other treatments. Yeah, so we try to mainly focus on hematopoietic organs. So we don't target disease. We target the innate immune system and then try to steer it in a specific direction. And we have a very translational angle. Yeah, and so as I mentioned, I focused here on cardiovascular disease. We have done work in transplantation. We do a lot of work in cancer. And we're also, obviously in this COVID-19 situation, we're also looking at infection. And so the induction of trained immunity may have very serious benefits in preventing infection. So that kind of summarizes what this lecture was about. There's some people I did not highlight throughout the lecture that I'd like to highlight here. Bram Teunissen, who is an instructor in my lab in New York, who builds all these technologies and keeps the lab going in my absence. Johanna, she's lab manager, she works really hard, make sure also that things are going. And Jess Munich, who in addition to all the work that he does experimentally, he's also our what we say, public relations guy, I think.
He's a great communicator.
He's a great communicator, yeah. And so, but there's lots of people, obviously, who I should thank, but most of them are listed on this slide. And then obviously I always have to thank the funding agencies, because you can imagine this very expensive type of research. And thank you for attending this lecture, and I'm happy to answer questions.
Okay. Thank you very much, Willem. It was a great lecture as always. Okay. Are there any questions for Willem? Please use the raise hand function. So maybe I start off with a question. Willem, if you are targeting these trained immune cells, trained immune macrophages and monocytes, do they require different nanotherapeutic formulations than the normal macrophages and monocytes? Do they take it up better or worse because you have more glycolysis? Or?
That's okay. So inflammatory cell types tend to be more active. So, but like this is not something that we've studied in vivo. So then we're mostly looking now at the bone marrow, and just look for materials that accumulate very efficiently in the bone marrow. For example, in these heart allografts experiments that we do, we see very marginal uptake in the heart allograft, which is full of macrophages. So we see much better uptake in the bone marrow and also in the spleen. So that's what we focus on. But I would suspect that trained phenotype or more inflammatory type phenotype is more phagocytic, and therefore will take up more of our nanobiologics. But that's not something that we've looked into.
Okay, thanks. I think Matthias has a question.
Yeah, so I wonder, Willem, if you can generally comment on advantages and disadvantages of nanoparticles. If you look at how people are trying to target immune cells, it's, I guess, mostly antibodies at this point, blocking cytokines and so forth. So what's your view on this? Where are the chances and where are the hurdles?
Yeah, so it's very difficult to reach lymphocytes with nanomaterials, for example. And then you'll have to start functionalizing the surface of these nanomaterials, essentially with antibodies. Yeah, then you may as well leave. There's no need then to use the nanoparticle itself. I think for myeloid cells, it's actually super efficient, and let's say the traditional paradigm in that field, trying to evade the immune system, I think it's a pretty good approach to go after the immune system with nanomaterials. I mean, like you have your siRNA studies where you show that can also be very effective. Yeah, so I think that in relation to antigen presenting cells and other phagocytes, I think nanotechnology has been neglected for way too long, and it's actually a great opportunity. How this will eventually mature and into if there's actual clinical modalities, we obviously hope so. And Sai and I are doing our utmost to get there, but yeah, I think it's a great opportunity for nanomedicine.
Yeah. Thanks. There's another question from Ada and also from Isabel. Ada, if you want to start.
I was wondering about the time of the memory and how much, like you said that the trained immunity lasts a couple months in mice and a couple of years in humans. So how long do cells remember after you treat them with the nanoparticles?
Yeah, so like I've thrown in trained immunity, maybe in this specific lecture is a lot of innate immunity, and maybe there's a lot of processes completely unrelated to trained immunity. Yeah, so in mice, the effects are a couple of weeks and not a couple of months. And so that's a big difference. Yeah, I'm actually not like, what specifically, like if you would have to redo this, or if you can, how long these effects last, or what?
Yeah, I mean, do you have to continue treat?
Yeah, okay. So one approach is the prevention of the induction of trained immunity. So if it's an acute inflammatory response that lies at the basis of the induction of trained immunity, if you then focus on preventing the induction of trained immunity, there's not like suddenly it will pop up again. And so, for example, in organ transplantation, what we see is that we can treat the initial inflammatory response, and that kind of keeps the innate immune system, quiet. And then we also don't see subsequent T cell activation. And so that's a one-time thing, and that has very long-lasting benefits. The induction of trained immunity, which finds application in cancer and infection, that's an effect that wears off. We haven't done these experiments. I think it's also beneficial. So let's say you're cured from cancer. It's kind of nice that your immune system goes back to normal. But we haven't yet done these experiments, but we have done experiments in different settings where we try to do some kind of maintenance therapy. So one of these studies that I presented that we published in Science Advances, we try to see if we can do kind of an induction therapy with the Simvastatin nanobiologics, and then do oral statin therapy to see if we can keep inflammation low. But you can imagine how many combinations you can come up with. For treatments, or let's say you could go and do it every 2 weeks or every month or whatever it is. So yeah, we do a little bit of that, but not for super extensively.
Thank you. Isabel, go ahead.
Oh, thank you, Willem. Outstanding, really fascinating talk. As a clinician, I'm a bit curious. I mean, which side effects do you envision from these particles? They look wonderful and perfect to me.
So the material itself, so that's your ApoA1 and some secret mix, and that's benign. And you can dose really high, and you probably have seen all these studies where ApoA1 HDL injections to kind of vacuum clean the vessel wall, which works in preclinical models, which does not seem to work in patients. But the amounts of ApoA1 that are administered in these studies, we need maybe like 1/100th of that. And so there you see that we're in the safety regimen, we're doing really well. But once you start introducing, an immunomodulatory compound, for example, something that induces trained immunity, you have to obviously be very careful, because before you induce a cytokine storm. But it's by design that they induce an immune response. So that's the other side of that coin. That's what we do in cancer. But yeah, thus far we see the safety profile is good. And by the safety profile, I mean, it's like at the dose we see therapeutic effects, the side effects are manageable, are within, what's acceptable. But it's that's mice and in primates. We don't know yet in humans. But
how far have you come with the human approach?
We're getting there.
Or can you reveal anything? Or
no. So we started a company, obviously, with the goal to. I can reveal that our seed we raised 6 million, and so now we have a little start, an inception. Yeah. We're one year into it. And we hope to to be in humans in maybe 3 to 4 years from now.
Yeah. Fantastic, lovely. Keep up.
Thank you.
It's very promising. Are there any more questions for Willem? No, if not, then I would like to thank everyone for joining us. Next week, Matthias will be speaking and joining us, and I hope you will all join us next week. And thank you very much, Willem, for your excellent talk.
Yeah, thanks everyone. I appreciate it on a Friday afternoon. Thank you.
Thanks, Willem. Great. Thanks a lot, everyone. Bye.
Bye.